Can everyone hear me? Great, awesome. We'll just get started in a minute. Are you able to see my screen? Yeah, that's a good question. Whether I can't see you and hear you, I'm trying to figure that out. There might be a Q&A section. I can see your chat windows. Let's see. Yeah, this is the first time I'm using Zoom in this mode. Let me see. Yes, I'm able to see the Q&A, but it looks like, I guess I can't see the videos of other participants. Okay, let's see. Let's see how this goes. At the end of it, we could try something else. Great. I can see everyone's chat windows, and I can also see the Q&A sections. For now, I'm not sure how to turn on the videos for the participants. So what we're going to do is, if you have any questions, just paste them either in the chat window or preferably in the Q&A window, which is right here. Okay. So yeah, we've got a bunch of stuff to cover. So I'm just going to get started. Move this away. Yeah, so thanks a lot for participating in this challenge, the ENCODE Imputation Challenge. Today I'm going to present a webinar on the challenge and introduce you to the core components. As you know, the challenge is hosted on Synapse, and this is the URL for the Synapse site. We've also managed to get generous prizes from Life Epigenetics. We've sponsored 3 prizes, $5,000, $3,000, and $2,000 for the top 3 winners. And also thanks to Amazon Web Services for sponsoring $100 worth of cloud credits for the first 50 participants. So let's jump right in. This is the outline of the webinar. First, I'll just introduce the challenge organizers, and then I'll introduce the challenge motivation objectives. I'll introduce the challenge data sets, the scoring measures, the timelines and rounds, incentives and rules, and finally, we'll end with an introduction to the Amazon S3 data challenge bucket, and also some instructions on how to use the AWS instances for compute. And finally, also a Q&A. So let's just start off with the challenge organizers. This has been a collaborative effort from several folks from the ENCODE consortium. I'll introduce the consortium in a bit as to what we do. But we've got Bill Noble from University of Washington, myself, Anshul Kundaje from Stanford, Manolis Kellis from MIT, Ziping Wang from the UMass Medical School, Carlos Boy, who's a graduate student in Manoli's group, Seth, who is part of the ENCODE DCC at Stanford, Jacob, who's a graduate student in Bill Noble's group, Jin, who's a software engineer at the ENCODE DCC and in my lab, Greg, who's in Ziping's group at University of Massachusetts, Jason, who's part of the DCC, and Gurkhan, who's a postdoc in Bill's group. So thanks to all of these folks who helped organize the challenge. If you have any questions about anything, feel free to post them on the discussion group, and one of these folks will help answer any questions. All right, so let's just get to the challenge introduction, motivation, and objectives. So I'm going to give, for those of you who are not that familiar with genomics, if there are any of you on this webinar, I wanted to give a really quick 101 on what data sets what these data sets look like and what the challenge actually is trying to address. So the human genome, as you know, is a long string of 4 nucleotides, A, C, G, and T. It's about 3 billion letters. And since the sequencing of the human genome, one of the central questions has been trying to understand the function of the genome, that is what every nucleotide or every word in the genome does. And when you're trying to interpret the genome and understand its function and its effect and the effect of mutations to the sequence of the human genome, cellular context really matters. And what I mean by that is, in any particular individual, in the human body, you have thousands of different cell types and tissues. And in any single individual, all these cell types and tissues typically approximately share the same genome. So you have the exact same DNA sequence in all of these cell types and tissues, and yet you have an incredible diversity of function, morphology, and so forth. And so, one of the key components of understanding genome function is to understand it in the context of these hundreds and thousands of cell types. So if you think about how we can actually start to figure out genomic function, we have to first begin by trying to dissect the anatomy of the human genome. So let's try to look into what are the different types of functional elements in the genome, that is, pieces of the genome that code for important things. So if you take the genome and you sort of unravel it from the nucleus of the cell, it would, maybe in a cartoon sort of way, look like this. So you got the DNA right here. And the most sort of well-known functional regions of the genome are genes, as shown right here. So genes are pieces of the DNA that code for proteins. That is, when you read the sequence of the gene, it can exactly be translated into a protein, and proteins do various important functions in cells. Now, what's interesting is the DNA is sort of not sitting naked in the nucleus. It's wrapped around these structural proteins to form what are called nucleosomes right here. And these nucleosomes provide not only structural stability to the genome and the DNA, but they also have important functions in regulating activity of the genome. What that means is in different cell types and tissues, different subsets of genes are activated and repressed. So an active gene will produce proteins and a repressed gene will not produce any proteins. And the precise combination of genes that are activated and repressed gives rise to a cell's identity and its function. Now, the other thing is that genes only occupy about 1.5% of the genome. The protein coding part of the genome is just about 1.5%. And so what happens is the remaining 99% of the genome has a variety of these various kinds of regulatory control elements. And the role of these elements is to essentially control gene activity. So these are little pieces of DNA, and based on their activity, a gene will be turned on or off. So think of them as like control switches for the genome. Now, to try to map where these elements lie and their functions and the activity levels in different cell types and tissues, we can start performing various kinds of sequencing experiments that can map biochemical markers associated with these different types of elements in different activity states. So here I show you a black triangular protein. It is binding these various positions of the genome. We can perform a particular type of experiment called ChIP-seq that can allow us to identify the precise locations of where this protein is bound. Similarly, there are a variety of other kinds of biochemical markers of function shown right here. These are often referred to as histone modifications or epigenomic markers. And these markers are biochemical markers added to the tails of these nucleosomes or to the DNA itself. And once again, you can perform sequencing experiments that give you sort of these genome-wide readouts of where these markers exist. So, these colored flags represent different kinds of markers. And as you can see, different kinds of markers mark different types of elements. So active genes are often marked by one type of marker. Active control elements are marked by another combination of these markers. And here's a repressed region of the genome that is marked by this gray marker, but not this one. So by mapping these various biochemical markers across the genome, we can start sort of annotating the genome and understanding genome function. So from a very sort of computational standpoint, what does this look like? A biochemical marker signal track across the genome is simply a vector of numbers. And these numbers effectively represent some sort of enrichment or statistical significance of enrichment of this measured signal at every position in the genome. Okay, so 3 billion positions in the genome, each of them will have a real valued number. The higher the number, the higher the sort of the signal of that element. So here's a nice example of what one such biochemical marker or an assay looks across the entire genome. This is a small portion of the genome, in the horizontal direction, and each row here represents a different cell type or tissue. So as you can see, there are some brain tissues here, there's B cells, there's liver, pancreas, and this is one type of biochemical marker. And you can see the same genome sequence exhibits incredible dynamics of activity of the same marker across different cell types and tissues. So different elements, different genes are being activated in different cell types and tissues. And so just as the Human Genome Project mapped a reference human genome sequence, now there have been large-scale efforts to map these various kinds of biochemical markers and their dynamics across hundreds of different cell types and tissues. So the ENCODE consortium is an is a consortium funded by the National Human Genome Research Institute. It's been active for over 10 years now. And the primary goal of the ENCODE consortium is to kind of obtain a comprehensive map of various kinds of functional elements in the genome using a biochemical profiling strategy. So as I showed you before, there are hundreds of different biochemical markers that you can measure across the entire genome in hundreds of cell types and tissues. And ENCODE has been systematically profiling these different markers across the genome in different cell types. So what you end up with is a giant three-dimensional data cube or a tensor, where one axis is genome coordinates, the other axis is different kinds of biochemical markers, and the third axis is cell types and tissues. And of course, this data cube is massive, and it's essentially practically impossible to completely measure it experimentally. So there are lots of missing holes in here. Typically, there are missing entire genome-wide tracks. So some assay is missing in some cell type across the entire genome. And so the main objective of the ENCODE imputation challenge is to build computational methods that can take existing measured experimental profiles of various assays in specific cell types and tissues, and then impute the missing profiles of missing assays and missing cell types and tissues. So that's the main objective of the ENCODE imputation challenge. So just to summarize the objectives, we will be providing participants, we have provided participants with a subset of the ENCODE data cube with missing tracks for specific assays and specific cell types. The participants are asked to develop methods to impute or predict missing genome-wide signal tracks. ENCODE will then perform experiments for the missing tracks as the challenge proceeds. And so in a truly blind prospective fashion, we will then evaluate the efficacy and performance of these various imputation methods in terms of how they recover experimental ground truth of these genome-wide assays. So just an overview of the Synapse Wiki. We've extensively documented various aspects of the challenge. Just a quick snapshot. So you should definitely check out all of these sections. Section 2, all the subsections in section 2 introduce the challenge, and section 3 is really focused on how you register, how you form a team, how you access data, how you submit predictions, what are the various kinds of prediction scoring metrics. And then the files tab has all the data, and the discussion tab has the discussion board that you should use to communicate with each other and with us. All right, so really quick overview of the challenge data sets. We are obviously not going to give you the entire ENCODE data cube. It's way too massive to wrangle within the parameters of an open challenge like this. So we've selected a subset of the data, which we call the imputation challenge data cube. Here I just show you 2 of the axes. Think of the genome going into the screen. So we've looked at a collection of 35 assays. Each of these assays is given an MXX ID, as shown right here. So each assay has an ID and a name. The first 2 assays here, ATAC-seq and DNA-seq, these are referred to as chromatin accessibility assays, and the remaining assays from M03 to M35 are known as histone modifications, and they have these H2, H3, and so forth names. So these are the assay names, and these are the corresponding numeric IDs. Similarly, we have assays and cell types. So we have 51 cell types and tissues. They're referred to by the CXX IDs, as shown right here. So C01 to C51. As you can see, there are common names for these tissues and the corresponding IDs. So here's adipose, adrenal gland, and so forth. All right, and as you can see, each of these assays, each of these cell types is labeled with 3 values, T, V, and B. I'll get to that in a second. That refers to training, validation, and blind test set. We will not be using the entire matrix. So some of these assays will be unused. Some of these samples will not be used for the purpose of the challenge. That's again just to make things tractable. So let's get into the training, validation, and blind sample test sets. So each of these colored samples in this data cube, in this matrix, are labeled as training, validation, or test. Training samples are typically marked with a T in this metadata table. Validation data sets are marked with a V, and the blind test data sets are marked with a B. So each of these represent each combination of assay and cell type. As you can see right here, C01, M16 has a corresponding file associated with it. This is a genome-wide signal track, a vector across the whole genome, corresponding to the activity values. This is in a standard format called BigWig, which is a binary format. You should look at the wiki for more details, and you can also click on this link as to how to read and write BigWig files. So we provide these BigWig signal track files for the training samples, which are combinations of assays and cell types. And we also provide them for the validation samples. The blind held out samples are prospective samples, so those we do not actually provide, and we will not be providing throughout the challenge. Those will be generated actually by the consortium during the challenge. And so we can really look at how the methods perform in a prospective way. So let me define what these training, validation, and test sets are. So as the name suggests, a training sample is a genome-wide signal track, which corresponds to an assay cell type combination for which data is available and the participants should use to train models. So what's the difference between the validation set and the blind held-out test set? So the validation set is also provided to the participants. You can obviously see these. And the point of this providing these validation data sets is that participants train models only on the training set, the data sets in the training set, and try to impute signal tracks in the validation samples. All right, so you should pretend like you do not have the validation samples, and you just use them as if they were a blind held-out test set. But of course, there is a likelihood that some participants may try to optimize and may try to overfit to the validation set. So we're not going to be using the validation set to actually declare winners at the end. This is primarily to provide sort of live feedback during the process of the challenge. All the participants will be submitting predictions on these validation samples, and we'll be using the validation samples to provide feedback on relative scores, rankings, and so forth. No feedback will be provided on the blind held-out test sets. These will be only provided at the end of the challenge, all the way at the end. It's the performance on the blind held-out test sets samples that will be used to declare winners. So that's the difference between the validation and the test sets. We could have potentially held out and not shared the validation sets, but we felt that they were available online anyway, and people might look at them anyway, so we just provided them as is. The nice thing about this is you can actually perform the scoring on your end and simply submit the scores rather than the entire tracks during the live updates of the rankings, and that makes the whole process a lot easier. But I just want to reiterate that when you're trying to submit predictions on the validation set, you should only be using data sets from the training set and pretend like you essentially haven't seen the validation set. So avoid overfitting to the validation set in any way, because you might appear like you're doing really well, but you might actually end up doing pretty badly on the blind held-out test data sets. Okay, so that's the difference between these three types of data. How do you get these data sets? If you go to the Files tab of the Wiki, you will see a variety of different files. You need to be signed into the Wiki, and you need to actually have registered for the challenge to see these data sets. Okay, so there are three directories. There's a submission template, which we'll get to later. Here's the training data. These are the big files for all the training samples. Here's the validation data. These are the big files for all the validation samples. And this FASTA file right here is the whole genome sequence. So we are going to be doing everything on the GRCH38 assembly of the human genome. Now, you can directly download these files from the Synapse site. So you can click on any of these files and download them one by one, or you can queue them. If you actually click on download options here, there are ways to queue files into a queue and then download all of them simultaneously. That's how you can do that from the web. If you prefer doing this from the command line, Synapse actually is quite nice. It allows you to do, has a very nice API that allows you to do uploads and downloads directly, either from a command line interface or through a Python or R library. So feel free to check out these links. Here are two simple examples of how you can download the entire training data sets and the entire set of validation data sets just through one command. Okay, so you can download the data either manually through the web-based portal on Synapse, or you can do it through command line or through R and Python. Now, what are the rules regarding the use of the data? Participants can only use data hosted or pointed to from Synapse. So you're not allowed to use any other data sets outside the challenge in any way, not even for like pre-training or fine-tuning. Okay, there's a question here that says, can you show how to load data sets into some Google Colab? We will try to do that towards the end. There's a bit of a session on the Amazon AWS. Hopefully we can get to that through this webinar system, but we will definitely address your questions as we get to it at the end. So participants can only use data hosted or pointed to from Synapse. You need not use all the data sets provided for imputation. You can use any subset you like. Obviously, using all of them will probably help you, but you definitely cannot use any additional data types. The only exception to this rule is features derived from DNA sequence. So we've provided, as I showed you in the previous slide, the whole genome sequence. You can manipulate the genome sequence how you like, represent it as schemas or use motif databases. Several resources are listed in the resource section of the wiki. So that's the only exception to the rule. That is, how you derive features from DNA sequence is completely open, but you really cannot use any other forms of data or any other data types that are not provided in the challenge. You can take the data tracks provided in the challenge and manipulate them in any way you like, but you cannot use any external data. Please read all the challenge rules carefully, and if in doubt, just feel free to ask the organizers in the discussion forum. So what are the challenge scoring measures? We use 9 scoring measures. There's a good reason for these. Prediction, as you know, prediction performance measures, each of them have certain advantages and disadvantages. Using just one metric often will cause issues. So we've tried to use a combination of measures, each of which is useful for different aspects of the data. So there are 3 global metrics. The first is mean squared error global, which is defined as a squared error between the predicted and the true values, averaged over all positions in the genome. It's regular mean squared error across the whole genome. We also have global Pearson correlation between predicted and true values, and global Spearman correlation, which is a correlation between the ranks of the values across the whole genome. So these are global measures of performance. We also have 3 local measures where we focus on specific regions of the genome that we have interest in. The first is mean squared error on what are called promoter regions. These are regions just 5' upstream of genes. We have the mean squared error over regions annotated as genes, and mean squared error over regions annotated as enhancers, which are regulatory elements that are often distal away from genes. So if you have independent annotations of what are promoters, enhancers, and genes, and we're going to be using those as well to compute these measures. And finally, we have 3 other measures. We have something called the mean square error var. So here what we do is, instead of having, so in the original mean square error, we simply compute the squared error for every position in the genome and then average it. Here we do a weighted mean squared error. So every position in the genome is weighted by the variance of that position for a specific assay across all the cell types. So more variable positions, for example, will be given sort of higher weight in the mean squared error. So it sort of focuses on the regions that vary a lot more than things that don't vary. And most of the genome is likely not going to vary as much. So this sort of focuses more on the regions we really care about. There's also mean squared error 1 observed, which is focusing on the top 1% of the true observed values. So these are mostly the regions that have real signal. And mean squared error 1 imputed, is also measuring the mean squared error in the top 1% of the predicted or imputed values. So we have 9 scoring metrics, and we need to figure out how to combine them to produce a unified scoring or ranking. So we previously used this ranking measure in a previous challenge, ENCODE Dream transcription factor binding challenge. And we're going to reuse that right here. So the basic idea is the final combined score is a sum over all measures, I, and over all assay cell type combinations, J. So the score is given by a negative log 10 of the minimum of 0.5 or the normalized rank of a participant. So we have a score, IJ, which corresponds to the performance metric I for a particular assay cell type J for a participant. So we compute the rank of that score divided by the total number of participants who submitted tracks for that particular assay cell type combination J. So this is a normalized rank. We take the minimum of 0.5 or the observed rank, and then we take the negative log 10, and then we average this across all the importance metrics and all the cell types and assays combinations. And all this is done actually over 10 bootstraps. This is to make sure that we take into account uncertainty of any prediction metrics. So what we do is we, for every assay set type combination in the test set or the validation set, we take 10 bootstraps. That is, we randomly sample 90% of the genome. We compute these performance metrics on each bootstrap, and then we aggregate them in various ways. I can't go into the extreme details of exactly how this is done, but we've detailed it extensively on the wiki. So please free to take a look at that. And we'll also be providing code for the scoring metrics and the ranking metrics. So what are the challenge timelines and rounds? The challenge essentially has started on February 20th and will extend until August 14th. We've decided to have 3 rounds. The first round is a competitive development round, which lasts from February 20th until May 15th. And then there's a collaborative round, and then a second competitive final round from June 6th to August 14th. And then there'll be post-challenge analysis following that. So I'll go through what each of these rounds mean. So the competitive round has already begun, the first competitive development round. So the idea here is that by the end of this round, you will be providing us with, you will be submitting imputed tracks on the validation samples and on the blind test samples. For the validation samples, as I mentioned before, you must only be training your models on the training data sets provided. And please, assume that you're training a blind, sort of in a blind setting. So really try to avoid overfitting to the validation data sets. When you provide these, what we will do is at the end of this round 1, the competitive development round, we will actually provide a ranking of participants on the validation samples and on the test samples. So we get a sense for the relative performance of the same participants on the validation samples that they actually get to see versus the blind test set samples they do not get to see. We're not going to be declaring winners after this round. This is mostly just to sort of help folks develop methods and see how they do. Also, there will not be live feedback during this round. So it's a development round. So you get to develop your methods all the way to the end of this round. And the last submission that you submit to us by midnight, US Pacific time on May 15th, will be used for scoring the validation samples and the blind test samples. So you will get 1 ranking on the blind test samples, but we will not be providing the blind test samples. Okay, so you'll just get your scores and your relative rankings relative to all the other participants on the blind test samples and the validation samples after this round. Now, after this first competitive round, we will have a collaborative round. This will be from May 16th to June 5th. During this time, we will have a virtual conference. We will have the top performing teams from the first round. They will be invited to present their methods and describe the lessons learned from the first round. The goal of the collaborative phase is to allow participants to share sort of their experiences, ideas, suggestions, and this really helps folks combine ideas and build even better solutions. So our goal is not just to sort of try to win this challenge, but also to come up with the optimal solution. And often the optimal solution comes by brainstorming and figuring out the best ways forward. So the collaborative round gives an opportunity for folks to exchange ideas and come up with even better methods. And in the previous challenge that we've run, we've seen dramatic improvements in the second competitive round after participants have had a chance to talk to each other and exchange ideas and information. So the final competitive round will again begin on June 6th and end on August 14th. This is a replica of the original competitive development round with one small difference. We'll be aiming to provide a live leaderboard for this round on the validation set. So again, you won't be getting rolling feedback on the test set. It's going to be completely blind and right at the end of this round as well. But for the validation sets, we will be providing a live leaderboard. So you'll actually get to see how you rank relative to everyone else. On the validation sets, again, there is a chance of overfitting here. So, take your rankings with a grain of salt. Also, really try to avoid overfitting or submitting, data sets or imputed tracks that have somehow seen the validation sets or overfitted to them. That way we get the participants really get a true sense for what the predictions rank relative to the others. So again, at the end of this challenge, we will be taking the last submissions for the imputed tracks for the validation and blind test sets, and by the final deadline, and then those will be used for the final scoring. So the winners will not be declared on the validation data sets. They will be declared only on the blind test data sets using the scoring and ranking metrics that were presented previously. And so in the end, a draft of the top 3 winners will be announced based on performance and relative ranking. Then we'll have the post-challenge analysis and announcement of winners. So after the final round, we expect all participants to provide detailed write-ups of their methods and share code, which allows the organizers to exactly reproduce the data pre-processing, training, and imputation. So this is critical. We really need everyone to provide reproducible results and provide detailed write-ups and code. You'll have enough time to do this. But the final list of top 3 winners will be announced after this validation round. And winning is contingent upon the organizers being able to reproduce the results with some assistance from the participants. All right? And the top 3 winners will be awarded the cash prizes as detailed in the challenge incentives. And then participants may consider working on their companion publications while the challenge organizers try to work on a flagship paper. So what are the incentives and rules for the challenge? The first incentive is, of course, we have actually a nice cash prize. Thanks to Life Epigenetics for sponsoring these. The first place will receive $5,000. If there are ties, the cash prize will be evenly distributed amongst the teams. There have been ties in the past, so that can happen again. Second place will be $3,000, and third place will be $2,000. And then for publications, there will be a flagship challenge paper. I'm not going to go through the details. We've documented, how this will work. All the challenge participants will be co-authors on the paper. The top 3 performers, assuming that they will provide reasonably significant contributions, will have significant authorship positions on the flagship paper. And companion papers, we encourage all of you to submit companion papers on your efforts. The companion papers can be submitted and published before the flagship paper is submitted or published, but we just need you to take explicit permission and notification from the challenge organizers. Also, the companion papers have a restricted scope. So the point is that the companion papers focus more on individual methods, and they don't really focus on global comparisons. That's what the flagship paper will focus on. But we don't have any restrictions on when you can submit and publish, as long as you restrict your methods, your papers to specific methods and results that you developed. And finally, the top performers will work with the challenge organizers to impute the larger ENCODE data cube. Okay, so we will really try to work with the final performance winners to help the consortium expand and impute data across the entire data cube. So finally, here's a discussion board, as you know, for all Q&A. Just a request to please be polite, courteous, and also try to be patient. We will obviously try to respond to all the queries as soon as we can, but do note time differences and so forth. And there's already been some active discussion, so feel free to talk to each other and also ask us questions if you have any. What's coming soon? There are a few things we need to release in the next 2 weeks. One is an exact replica of our scoring scripts so that you can know exactly how we are scoring your predictions. And we will also be providing exact instructions for submission of the imputed files for validation and test sets. This should be pretty easy, at least for the first round, because we're only going to have you submit these towards the end of the round, and we'll be using the last submission as your final submission for scoring. In competitive round 2, we will activate a live leaderboard with weekly feedback. So it's not going to be instantaneous. It takes a bunch of time to compute metrics and rankings. So it'll be a weekly update. Specific day in the week, we will take the last submission of each participant and use that for the rankings. And the exact instructions for submissions, and again, will be provided at that time. So that's it for the introduction for the challenge. We were then supposed to give a brief introduction to the Amazon AWS and S3 data bucket. So I'm going to try to see if I can add I can add Rob as a presenter for this meeting, because I don't think anyone else is able to present. Sorry for these technical difficulties. In the meantime, maybe some of you could post any questions that you have on the Q&A, and some of the organizers who are on the chat can also help answer some of these questions. I will try to figure out in the meantime how I can get Rob and Amanda to present the video. Oh, I see. Okay, this is great. I can allow to talk. Rob, are you there? I think I've given you speech permissions.
Yes, I believe so.
Can you say something again?
Yeah, this is Rob.
Great, awesome. Can everyone else hear Rob? Just type yes if you can. Okay, great, fantastic. Okay, so Rob, what I'm going to do is I'm going to have to run the slides, but at least I guess you can see the slides and I'll just move them ahead as you want me to. Does that sound fine?
Sure. I was going to do
Okay.
some live presentation, but we can just work with the slides.
Oh, that's a good point. Okay, let me see if I can.
It's really.
Are you able to share your screen? Do you have an option to share your screen?
View options. No, I do not see an option to share my screen. So we can just work with the slides.
Okay, maybe we can do a screen capture sometime later and post it or something if we need to. All right, let's just run through the slides for now. My apologies, everyone. We tried to set this up as a webinar instead of a chat. I think it would have been way easier to just do this as a conference, video conference than this webinar thing. Okay, so we'll continue with the AWS presentation. So go for it, Rob.
Okay, so you can hear me okay?
Yeah. Okay,
great. So the first slide is how to skip this presentation. And I'll just say really quickly that if I'm not sure how many attendees we have online here, but if you're not familiar with the Amazon cloud, it's essentially a large collection of computing resources and servers and that has excess capacity that they make available on a rent by the hour kind of basis. That's pretty. And so what we're talking about is two things in this short presentation. First, we're talking about how some of the data, or the data for this challenge is stored on the cloud and how you can get it. And then secondly, if you were to take advantage of the credits that are available to you, $100 in credits, and to continue to explore using the cloud, it can be used as a computing platform. So I'm going to just sort of describe both of those aspects. The reason this says how to skip this presentation is if you just want to get to the data, there are two things you can do. You can install something called S3 browser, and that is a application that runs on your computer, and it connects to the Amazon cloud, and it connects to one of the first of four different jargony terms that I'll introduce here. And then you can just simply use the S3 browser application as a way of looking into the storage bucket. It's called ENCODE Imputation, and seeing what's there. And then you can use that to download files. The files tend to be 300 megabytes on the small side to a gigabyte, and you can generally download those over a reasonable link in a matter of a minute or
Rob, I'm sorry, I think I I just upgraded you as a panelist, so I think you can actually now share. You should be able to share your video and also share your screen. Can you hear me, Rob? Oh man, okay. Hey Rob, can you try connecting again? Can you try
Okay,
just
now
try
I'm back.
connecting back? Oh, great, awesome. Okay,
Yeah.
so you should be
Oh,
able to share your screen if you need to. Okay.
yeah. Okay. Let's see. Where was I cut off? Probably I was trying to explain this slide. So I'll just
Yeah.
sort of begin over. If you're only interested in taking a look at the available files for the challenge, you can use the S3 browser application, and that will connect you anonymously to S3, and you can see what's in the ENCODE imputation bucket. And then you can even just use wget as shown here in this example command, where the command will download the file c03m16.bigwig. And the download, if you have a decent connection, will just like take a minute or so for a file. So this kind of implies that you could pull all the data down to your own computing environment. But the alternative is to actually work in the cloud. And so the rest of this presentation is going to be concerned with what it would mean to be on the cloud. Pause, stop recording. I don't know how to advance this slide. Oh, there we go. Okay.
I'm able to advance it, so I'm just going to be doing that.
Yeah, let's just stay with this plan right here. This is great.
Okay.
Okay. So on the Amazon cloud, obviously it's a secure environment that you're connecting to, and you have to be able to authenticate somehow. There are three different ways of connecting to cloud resources. So the first one is that there is in your browser, you can connect to the Amazon console. And we'll see an example of that. When you log in, you're either the root user, the first person to create the account, or you can also create additional users. And there you just simply have a URL that you connect to, and then you have a user ID and a password. The second way of connecting to a cloud resource is when you have a virtual machine, and the jargon term there is EC2. So the Amazon cloud has EC2 instances, which are computers, and they range in power from a Raspberry Pi to a supercomputer, like a GPU-powered massive machine. You pay, obviously, in proportion to the compute power, and you pay on an hourly basis. So it's a really good idea to know how much your machine costs and how to stop and start it so that you're not paying for it when you're not using it. And when you stop it, it still retains its state. It's just like kind of turning it off, and then you can turn it back on again. The way to connect to a virtual machine through SSH is using a certificate file. In this case, we have one called myec2certificate.pem, and you have to change its permission to 400 in Linux before the SSH command will work properly. And then you just simply say SSH, and then you give a username at the very far right there, ec2-user at, and then I just give an example IP address. So that would be how you would SSH into your Amazon cloud machine using the certificate file. And then the third way of accessing the cloud is through something called an access key. And I set this aside as something that you can learn to use, but there is a certain danger associated, which is that access keys are your way of connecting to the cloud and starting up resources. And we've had problems with people who work on the in the GitHub environment who end up leaving their access keys accidentally on GitHub, because what happens is there are bots that will scan GitHub for access keys, and then once they get them, they'll log into your account and they'll spin up resources and start mining Bitcoin, and it costs an enormous amount of money, and then you have to go to Amazon and say, oops, somebody got my access keys, and can I please be forgiven the $15,000 that I owe? And that $15,000 is just a random number. It's not from an actual example case. No, that's not true. Actually, I did that. So the Amazon command line says here, CLI, command line interface, is something that you can install on a machine and use to talk to the Amazon cloud. And when you do that, you can, for example, use the AWS S3 command, but it will require you to authenticate by using your access keys. So that's the context here. Okay, let's move on to the next slide. I have a lot of slides to go through, but they're really intended as a resource. So the idea would be to say, here's the script of what we're going to do, and here's the slides that show you doing that. And as you're following along, don't worry if this kind of all flies by too quickly, because with a limited amount of time here, really we just kind of want to introduce the ideas and then know that you have this resource available. And you can also get back in touch with us. I run a consulting group here at the University of Washington, and we're interested in enabling you to be able to use the cloud. So the script goes as follows. You log into the Amazon console as root. That's once you have your account set up. You create a user, which has some kind of credentials. You then log in as that user. That's where you do all your work. And you set up an EC2 instance. That's a virtual machine. Then you connect to that virtual machine using SSH, as we just described. And then you would test out and configure the command line interface there. And then you would use that to copy files from the ENCODE data source into your local directory, where you would then run your software to analyze it in the due course of pursuing your entry in this competition. And then you would finally return to the console, that's the thing in your browser, to stop that instance when you're not using it, so that you're not being charged by the hour for it, particularly if it's powerful. How much is a powerful instance? You can pay as much as $25 an hour if you need to use a GPU, or 4 GPUs at a time, actually. But the lighter machines, like the one I'm currently running for this example, cost like 8 cents per hour. So that's the kind of the range of costs. You can get a pretty powerful machine for $1 an hour. Let's go to the next slide. Okay, so now we're going to see a bunch of slides, and I'm going to say next fairly frequently. So this is just what logging into the console looks like. Next slide. And you'll notice that here I've connected to the IAM, that's Identity and Access Management, and it tells me that there are 10 users. So let's click on the users and keep going. And here there's a couple of the users are named Amanda and Anthony, but there's a button to add a user. So let's click on that and keep going. And here I'm not going to give this new user, this is a wizard that sets up the user. But I'm not going to give them programmatic access through the access key because of the danger I mentioned. That's something that you can generate later, but we are going to give them console access. Okay, keep going. And now here we add them to a group so that there's some kind of capability or permission associated with them. So the admin group means that this user will be able to do admin stuff, which is just about everything except delete the account. Next slide. You can tag the user by giving a project that they're associated with. This helps in account administration. Let's go on. And then finally, we just simply get to the end of the wizard and hit create user, and that's going to work because this is slides that I've already set up. And when you set up that user, here's the user. I give the user the name Fred Johnson, and there's a file that you can download that's the Fred's credentials to log into the console. So you can see that there's a username down at the bottom in the big rectangular box. There is a password, which is Fred's first login password. Then there's an access key ID and a secret key, and those are those commas with nothing there. That's the dotted oval. You'll notice that there's nothing there because we didn't ask for access keys from the get-go. And then finally, there's the HTTPS, UW, CloudZar, et cetera. That's the link you go to sign in. Okay, let's move on. Now, this is the slide that I created to warn you about access keys and alternative ways of getting to the ENCODE data. So I'll let you read this on your own time to save time here. But again, the main thing is that you can use the S3 browser. You can install the Amazon command line interface as shown, or you can use the Boto3 Python package to talk to S3 object storage. So a quick note, when we say S3, that's Simple Storage Service, that's the Amazon object store, and it's not like a disk drive. It looks a little bit like a disk drive, but it's not files that you can open and then index into that file. Rather, it treats the files as objects that have URLs, and then generally you will copy them into your working environment where you can actually open them and access them across the file. So but the S3 storage is much cheaper than using storage that's attached to a disk drive attached to your virtual machine. Okay, so let's move on. Next slide. Okay, so now Fred Johnson logs back into the console here and finds the place where it says launch a virtual machine. So that's the second bit of jargon. EC2 is the Amazon virtual machine. So this is what you click on as Fred. Keep going. And now here is another wizard. And again, this is in the Amazon console in your browser. And it's simply going to walk you through these at the top 1, 2, 3, 4, 5, 6, 7 steps. But we're just going to jump ahead by using the review and launch button. So you'll notice first big, huge oval here is, oops, let's go back. Amazon Linux 2 AMI. So this is a image of a computer with an operating system that's an Amazon version of Linux, and I'm choosing that. It will have the Amazon command line already built in. And you'll notice that I'm choosing the general purpose M5A.large machine, which sounds pretty impressive, but that's only 8 cents per hour. And then I'll click on the review and launch, so let's move onwards. Okay, so after going through this sequence of steps, we simply launch the instance, and you'll notice that at this point, we talk to the Amazon interface to get a key pair file. That's the certificate to allow you to log in, and we're going to call it Fred Johnson ENCODE, and that'll be with a .pem file extension. We use that to log into the machine from our own machine. Okay, next slide. And then here in the process of spinning that up, the console will create an entry for the new machine. And you'll notice that it has a green dot up at the top indicating that it's running. And if you keep on scrolling over to the right, it continues with the second row down here, and it gives you an IP address. So the point is, Fred is logged into the console. He started up an EC2 machine. That's a virtual machine on the Amazon cloud, and he has an IP address. Okay, let's keep going. So while that machine is up and running, let's go to the next slide. And now Fred opens up a bash shell, and he copies the Fred Johnson ENCODE file, he chmods it to 400, and then he does an SSH command, which connects to the machine. And the text in the lower part of this box here shows logging into that machine, including the suggestion that you run sudo yum update to update the operating system. Let's go to the next slide. Okay, no, that's previous, next slide, there we go. And then immediately after he's logged in, Fred types wget, and there's the rest of that command there, and that takes a few seconds and it grabs the C03m16.bigwig file. So if we had to stop this presentation here, that's just enough to show you that you can get a machine on Amazon and you can wget one of the ENCODE competition files called CO3M16. So there we go. Let's continue onward until you guys interrupt me with we're out of time. How's that? So the remaining tasks in my little script are to use the CLI to copy an ENCODE data file. That would be a second way of doing it. And then return to the console to stop the EC2 instance and make an AMI. Okay, so then terminate the instance. So let's keep going. We'll just spin right through this stuff. So you'll notice at the next line, I'm still on that Amazon machine, and I typed the command AWS, which is the AWS is how you invoke the command line interface. And we type in configure, then you have 4 questions to answer. And I've already answered the first 2, so it has the defaults all set up. Those are the access keys that I warned you about. And then there's the default region, which is US West 2. That's where this S3 bucket happens to be sitting. And the output format we don't care about. So once you type in these things, now the AWS command line interface is configured, and it will be able to do stuff on your behalf. Okay, let's go to the next slide. You can read that stuff later. Let's see, what is this doing? Okay, so this is saying AWS S3 ls, and then it gives the name of the bucket. And it tells us, well, in this bucket, there is a submission template folder, a training data folder, and a validation data folder, and then there's a file there. Okay, so it worked. Let's go on. Again, this is from the EC2 instance, the Amazon virtual machine that we're logged into. And so then I did the copy command, and sure enough, the copy command grabbed the bigwig file, and it's identical in size to the wget file. So they must be the same file. Let's keep going. And then here we did S3LS, and the idea here is to show you getting the contents of those ENCODE imputation bucket folders into a file called foobar so that you can decide which of these files you want to grab. Let's keep going. Okay, now we're back on the console because we've done a bunch of work on our EC2 instance and it's time to stop it. The first thing I'll point out is that the instance has a name, but nobody's filled anything in. And this is a bad idea because it becomes a zombie machine and nobody knows who's using it for what. And that can end up costing money. So you want to have in mind that when you run on Amazon, the Amazon cloud, you want to have some cost management practices in your mind. And one of them is to name your instances so you know what they're doing and whether or not they should be stopped or deleted. Okay, let's go next. So Fred Johnson clicks on that little pencil and edits his instance name, and now it's called Fred Johnson ENCODE, and that's much better. Let's move on, next. And now here we are on the console, and we have clicked on the actions, which is the button at the top center, and that brings us a drop-down menu, which has instance state, which has a further drop-down that gives us the stop and terminate options. So stop is how you stop the machine, and that's the way you end up not paying however many dollars per hour for it, and that's a good thing to do at the end of the day. And then terminate actually just blows the machine away, so everything there will be lost forever. Let's continue onward. And then the other thing that you can do with your machine is, since you've done some work on it, and let's say you want to stick it on the shelf so that somebody else can use it, you can create an image of that machine. And think of an image as just a big, huge byte capture of the entire state of the operating system, all your code, all your data, everything gets bundled up into this big file, like a zip file. And that's how you do it. You go here and you say create image, and that will create that file. And then you can set the permissions on that and share that with your collaborators, for example. So you could have 17 versions of this same operating environment up at the same time because people will keep spinning up this AMI. And not only that, but it's kind of cool. You can develop on a cheap machine and then you can spin it back up on an expensive machine when you actually need to do the processing. Okay, next. A few final remarks. These are on cost and strategy and so forth, and the fact that you can move data around inside the Amazon cloud at no cost. Particularly if you keep everything in that same region in Oregon where the S3 bucket is for this event, for this competition. But when you pull data down off of the cloud using your account, then that's going to cost somebody some money, 10 cents per gigabyte. So that's everything. I'm hurrying so that we can get through everything. And there we go, questions.
Great, thanks a lot, Rob. So it looks like I can actually give folks permission to ask questions. Let me just start doing that. So if you would like to ask a question, can you just, oh, there are a few questions already. So let me answer the questions that have been typed out, and then I'll give you all a chance to also ask questions through actual voice. So I guess the first question is, can you show how to load data sets into a Google Colab notebook? We can possibly send you some instructions on that. It'll be too difficult to do it now live. So we'll get back to you on that. Himanshu. Anonymous attendee says, will you provide extra information about the bigwigs? Example, which lab generated it, lab protocol, read length, antibody. Good question. Yes, we can provide this information. We will. Dave asks, use refgene to get coordinates of exomes. Yes, since it's based on sequence, if you're just using it as a manipulation of the sequence itself or some annotation on the sequence, that should be fine. Chris Mancuso asks, what is the total size of the training and validation files in terms of gigabytes? I don't have the exact value for that, but we will get back to you on that. We will post the total files. Sean Mahoney asks, can we assume all training data is already normalized and on the same scale as the expected imputed data? That is, are we provided with read counts or some transformed value? Good question. So what we're actually providing is a sort of normalized recount. We are providing the negative log 10 of the p-value of the signal. So this is the negative log 10 of the statistical significance as estimated from a Poisson model. They are reasonably normalized. There are obviously potentially issues with like differences in data quality can result in slightly different ranges of scale of the negative log 10 p-values, but we will treat them as is. So you don't have to explicitly renormalize it in any way unless you want to. The blind head out test set files will also be processed in the same way and will be evaluated against using the negative log 10 p-value signal values per base pair. Sorry, I should say the evaluation will be happening at 25 base pair resolution, even though the current tracks are provided at 1 base pair resolution. There's more details on that on the wiki, so feel free to check it out. But it's a good question. Briefly, to summarize what I just said, they are in some ways normalized. They're normalized against a genome-wide background and based on the Poisson model. So the signal tracks are negative log 10 p-values, if that helps. Okay, so if others have questions, you can feel free to, will the evaluation include a transformation? Bill Noble's lab likes applying the arc-SiNH transformation, for example. So these are already transformed values, the negative log 10 p-values. So we won't be actually transforming them again in any way. These are already pre-transformed. Any other questions? You can feel free to type them out, or just type your name or something on the Q&A, and I can give you speaking permission. Okay. I'm guessing the submissions will be in bigwig format. Do I have to provide bigwigs at 25 base pair resolution, or can I just provide it at 1 base pair resolution? We're going to give us, there's actually a submission template, and we'll give a lot more instructions on how to submit. The submission template is at 25 base pair resolution. So the reason is twofold. Firstly, the signal is not actually informative at a 1 base pair resolution. So 25 base pair kind of works out reasonably well. And secondly, 25 base pair bigwigs are far more compressed, so it makes it much easier to upload and download files. And that's the reason we're primarily evaluating at 25 base pair resolution. So the submission template provides chromosome start stop, and you just have to fill in values. So we expect you to provide an exact same submission template. There's a bed graph and a bigwig submission template that's provided. So you can use those to effectively generate your bigwig files for all the imputed tracks and submit them. So it will be at 25 base pair resolution. Any other questions? Okay, if not, I think we will end the webinar here for now. But you should feel free to ask as many questions as you like, and we'll get back to you as soon as possible. You can email us, or ideally use the discussion board. Emails, if there's something private that you need to ask, you can do that via email. Ideally, all discussions should be on the discussion board on the wiki. All right. So thanks again, everybody, for participating in the challenge. I think it's going to be a whole lot of fun, and hopefully we'll have some state-of-the-art methods by the end of the challenge. And thanks to Rob again for presenting his AWS session, and thanks to the organizers. Thanks to the ENCODE consortium for providing the data, and thanks to Life Epigenetics for the prizes, and Amazon for the credits for the top 50 for the first 50 participants. All right, so we'll end the webinar there. Thanks again, everybody, and hope to talk to you online soon. Bye-bye.